Hello everyone, welcome to Inspire short lecture series in science. Today we are going to discuss a very important topic of vaccination. Given the pandemic that's going on, this topic assumes quite a significance. In this particular lecture, we are going to look at a few points that relates to vaccine development and then a bit of history and the cultural aspects of vaccines and some science about some numbers about whether the vaccines are truly effective. First thing we are going to see is how is a whole virus vaccine produced, or what exactly is a vaccine, right? A vaccine is basically uh, it's something to trigger our immune response. So when a new pathogen, let's say COVID, enters our body doesn't even know how it looks, so it is not able to effectively fight it. When we give it a vaccine, it's like we are training our immune system to identify the virus. So when the virus comes in, we are able to effectively fight it. So one of the major steps in vaccine production is about whole virus production. How are we going to propagate the virus? There are many ways to do this. Nowadays, for example, in mRNA vaccines, you just need to know the sequence of the virus. You don't need to actually produce the virus. But some vaccines like Covaxin, which use a whole from Bharat Biotech, which use whole virus inactivated vaccines, they need virus propagation. And this is where much of the controversy is coming up today, so we will focus on this a little bit. What typically happens in when a virus enters a cell is the virus outside the cell is pretty much non-living. It's just a nucleoprotein crystal. It has, however, the protein contains something called a spike protein or a surface protein. It has a cognate receptor on the surface of the cell. It binds to the receptor, enters inside, hijacks the cellular machinery. It uses the proteins that are proteins and the food available inside our cells, makes copies of itself, comes out, and then the cycle repeats itself with the other cell. This is a very short primer on how we can actually grow the virus. See, if you are having a bacteria, you have a medium in which the bacteria can be put and it grows. For example, for E. coli, it's a it's called yeast extract, and then you add some glucose and you're done. You add some proteolysis proteins, you add some glucose, you add yeast extract, and the bacterium will grow. Whereas to grow a virus, you actually have to have the host cell. So what do? We take the host cell, we grow this host cell in large reactors, add the virus to it, let the virus grow inside the host cell, and we cannot now, the virus is so small, right? You cannot just separate it so effectively. So what we do is we separate it while it is inside the host cell. We use a technique called centrifugation. We use several other techniques, physical separation techniques, wherein the cell is completely removed from the medium. This is a very important part here, right? When we try to extract the virus, the first thing we do is we remove the infected cell from the medium in which it is growing, and then we take the cells, wash them, and then we do something called lysis. Lysis is basically breaking the cell open, and then we release the virus, and then it undergoes more purification steps, at the end of which the virus is finally heat inactivated or enzyme inactivated, and that is used to make the vaccine. So you can see from the point where the cell is grown to the point where the virus is actually used as a vaccine, there are a whole lot of steps and whole lot of purification involved. This is for a good reason. The virus the virus particles that are used here for vaccine production, this cannot have any traces of contamination from this point. If it has, it will be rejected. this cannot have traces of contamination. Let's remember that and move on to the next slide, which is a more important question. Do we use FBS? First thing, what is FBS? FBS is a very fancy way of saying fetal bovine serum, or it is the serum from a fetal bovine, like calf, right? Why do we use it here? Because of the nature of this eukaryotic cell. We have eukaryotic cell here, right? Now, eukaryotic cells inside our body, how do they grow? They grow because our blood supplies a lot of nutrients, and also it supplies some signals that are essential for the cell to grow. Outside, in a place like a petri dish where we actually grow these cells in the lab, we add, instead of our blood, we add fetal blood, calf blood, right? And this is used to grow the cells. The cells are grown, and then the medium is changed, and then the virus is infected into the cells. Then the cells are again grown, and then we go to the step what we discussed here. This comes after all of this. So you can see really from the point where FBS is being used to the point where you are actually growing the virus, there is again a lot of changes. And also, just as I told here, when we remove the cells from this medium and then we purify for the virus, there is not a trace of what is left here. There cannot be any trace of FBS used in this stage in the vaccine, right? Let's be clear, there is no FBS in the vaccine, right? So is there FBS present in the final vaccine? Big no. Big emphatic no. All media components are thoroughly removed through various steps. We remove all proteins, all media components, including FBS, and further steps of purification removes any trace amounts of media. If there is any impurity, it is unfit for clinical human use. This is the law, this is the regulation. So there is no FBS in the vaccine. Now, obviously, there are a few associated questions in this problem that come up. One is, why do we need vaccines at all? Why should I even vaccinate? In one figure, in one plot, I can tell you the importance of vaccines. This is the incidence of poliomyelitis. I don't know how many of you have seen a polio case. It is very, very rare today in the world, but in 1980s it was not too rare. Especially in India, it was pretty endemic. It was quite common in India. In 1980s, the world had about 400,000 or 4 lakh cases of polio infections recorded. In 2016, we brought it down to 46. In 2020, we had a mere 30 cases in the entire world. That's how much, from 400,000 to just 30 cases in 40 years. How did we achieve this? We achieved it by mass vaccination. Even today, India's oral polio vaccines, which are again inactivated vaccines, are a major success story in the human history. They are done at a scale that is unprecedented anywhere else in the world, and we have been able to save lives and our children's lives with vaccines. Okay. Now, another question that is often brought up is vaccination culturally acceptable to Hindus? This is a very important thing. See, there are some cultures which think vaccination is not acceptable to them culturally. Whereas it is not so in India. In fact, pre-Mughal, pre-British India was the world's pioneer in immunization programs. We had a social immunization program long before the Western world stumbled upon this wondrous thing called vaccines. Right? We had practice of doctors collecting human pustules from pox infected people, drying them, processing them for a year, thereby creating inactivated viruses and then using them to immunize people. This has been going on for centuries. It was stopped by the British, and then we had a whole bunch of epidemics break out, and then we adopted the Western vaccination policy. But still, we as a culture, we as a community were not averse to immunization. Our ancients knew the value of immunization. More important than that, vaccination is just an improved process of immunization, and we should have no hesitation in accepting vaccination. The last question I have is this: is vaccine for COVID-19 worth it? I have expressed it many times. The speed at which we have created a vaccine for COVID-19 is unprecedented in the world. Within a year, we have many vaccines that are vying for each other in the market space. But it's not all, it is not everything is a gimmick, right? They are truly effective. For example, look at this study from Israel. This gray bar represents the unvaccinated people. This is 100%, right? So whenever we take one of these bars, we compare that to 100% in the unvaccinated group. So let's take the severe disease. Severe disease means symptoms. There is a severity scale that we use to measure COVID severity. And on that, if we say severe disease, compared to unvaccinated people, this is just 26%. Three-fourths of the people had far less severe disease after just one dose of vaccination. How about hospitalization? A fraction of severe diseases get hospitalized, but compared to unvaccinated group, this is 28%. How about deaths? It's just 22% of unvaccinated people. So you can see here that compared to unvaccinated group, vaccines save lives, it saves hospitalization costs, and it saves you the discomfort of having a severe disease. So there is no reason for us to deny ourselves vaccines, especially when they are available. I hope this lecture helped answer some of the questions that you had on vaccines. And till the next one, have a good day. Vaccinate yourselves. Bye.